Hi everyone, my name is Thibaut Delem. I'm a student in Neurosciences in the EM Online Master in Bordeaux and currently an intern at Laval University. In this video, I want to share with you my work in Quebec by introducing you to SEM, an electron microscopy technique. So, I will talk about the work of Perez's team on counting synapses using focused ion beam scanning electron microscopy. But before going into details about the technique, let's talk about why they use it. As you may know, every healthy human being has a brain, and this brain is connected to the rest of our body thanks to neurons. However, they are useless if they are not able to connect with each other, and that's where synapse comes into play. So in this presentation, we'll focus on synapse, and especially on cortical brain synapses. First, a synapse is a structure composed of pre- and post-synaptic sites separated by a synaptic cleft. And to connect both sides, neurotransmitter will be released by the pre-side in the cleft and will bind to receptor of the post-sides, inducing a response. Then the signal can keep going. Finally, in order to become synapse expert, you need to know the two main morphological types of synapses in the cortex. The gray is type 1, asymmetric, with a thicker density on both sides, and the type 2, symmetric, with similar densities. Alright, now after I bored you with theoretical explanation about synapses, you might be thinking, what's the matter with those synapses? Actually, those synapses, because of aging and neurodegenerative diseases, tend to decrease. So being able to count them is a major step forward to understand their function and the impact of aging on neurodegenerative diseases on synapses. Enough talking about neuroanatomy now. Let's talk about the most interesting part, imaging. I will divide this part into two segments, with first a presentation of SEM imaging and then how it could be useful in our case to count synapses. Let's begin with SEM then. Also known as scanning electron microscopy, this imaging technique implies electron and will produce beautiful microphotographs. Forget all you may think about neuroimaging, the colorful and fluorescent pictures you've probably seen. Now we'll talk about black and white, or in fact gray, and trust me, you will like it. This technique, no matter how ugly it looks, is the most precise one. With a 2 million power magnification against 2000 in optic microscopy, you're pretty much able to see everything. I know, it's exciting. And you want to know something even more exciting? We can do awesome 3D reconstruction of synapses in our case. But the awesomeness has a price. In fact, all of those acquisitions will be produced thanks to a combined FIB-SEM microscope, basically a very expensive microscope. Built to emit an electron beam, those electrons can interact with atoms that compose the tissue and get back to the microscope to produce this type of image. This is what we call BSE, backscattered electrons. But to be able to reconstruct whole synapses, we need to have images related to each section of a sample. This is why we have FIB in FIB-SEM. The focused ion beam will be useful to mirror a sample at a defined thickness and allow the serial acquisition of images. Moreover, this system can be fully automated. An electron beam is emitted, BSE comes back to the microscope, an image is acquired, the FIB removes the sample surface already acquired, and we restart the whole process again and again. However, FIB has a dark side. It destroys the sample already acquired, which makes the study of the same sample impossible. But it's one drawback over many advantages. So, anyway, this is how they obtain their whole stack of images, what we call the 3D counting frame. Nevertheless, those images need to be processed, and to do this, Perez's team used different software. And the most interesting one is Reconstruct, which was useful to represent synapses in 3D. To do that, they trace contours of synapses on each section. The idea is to define for the software what is the object, the synapse, and what is the environment, basically everything else. Then, they could be able to identify synapses and even look at their organization. So, let's have a look at those serial sections, for example. You can see that the synapses, pointed here with arrows, has a specific organization called ring shape. The synaptic junction seems in one part at the beginning, then split to get back in one part in the end. Now, this is how it looks in 3D. Clearer now? Here you have the axon terminal, the synaptic junction, and here the dendrite. This approach helped them to understand that this synapses was a perforated asymmetric synapses, and this wouldn't be possible without this technique. Finally, let's talk about counting. In this paper, the quantification has been done using several methods. But to make this important part understandable, we will focus on only one technique, the direct study of serial sections from stacks. In fact, in this method, you have to define a box. Then, all synaptic profiles that are in the box are counted, and the ones that intersect with the edges aren't. Then, they divided the total number of synapses counted on each section by the volume of the counting frame to obtain the number of synapses per unit volume. This is how they obtain an average number of synapses per studied regions. So, as you saw, comparing with other imaging techniques, this SEM technique allows us to define many important features of brain organization and is a major tool to study brain's microanatomy. This offers great opportunities for the future to understand the impact of aging or diseases on the number of synapses, and also to get a clearer representation of brain synapses by using 3D. To conclude, it doesn't matter how ugly you look if you're just the best microscopy technique in the world. Thank you very much for your attention.